Hello, my dear students. Welcome you all to Sri Jaindra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today I am going to start with a very interesting topic which is basically related to neural control and coordination, sensory organs. In this topic, we will be studying about the types of receptors, all the types of sensory organs, eye, ear, skin, nose, everything will be discussed. So starting from the types of receptors, first we will study that what are receptors. The environmental changes, they are detected with the help of these receptors. The receptors, they are the sensory organs or a cell which receives stimuli which receives stimuli from the environment. Stimuli means the changes in the environment from outside or inside the animal, from outside or inside the animal, right? And then it subsequently passes the impulses to the nervous system and passes it to nervous system. So basically receptors help to connect the nervous system, which is a complex organ, okay, to the external stimuli, means the external environment. In its broadest context, sensation is the consciousness or the unconscious awareness of external or internal stimuli. Cells which are specialized to receive stimulation are called receptors. So receptors, they are the specialized sensory organs or cells which help to receive the stimuli from outside or inside. The sensory system consists of simple to complex structures called sensory receptors. An animal responds to a stimuli or the change in the environment in four basic steps. The first step is sensory transduction. Okay. The second step is transmission of that impulse. The third step is integration, means analysis of that impulse. And the fourth is the response, that is action. First is the sensory transduction, means taking or receiving of the change or the stimuli. Then the stimuli is transferred or transmitted to the nerve endings. Then it is analyzed by the different centrally placed structures like brain and spinal cord or the ganglias, and then the response is executed. Now we're going to discuss about the types of receptors. Receptors, they are classified on the basis of type of stimuli they receive or on the basis of type of stimuli they are getting. Exteroreceptors, they receive stimulation directly from the external environment. Exteroreceptors can be of following types: photoreceptors, which are sensitive to light, that is eye; thigmoreceptors, which are sensitive to touch; tectoreceptors, which are sensitive to touch; tangoreceptors, which are sensitive to touch along with pressure. Touch along with pressure means the touch, and that is not a simple touch, it is pressurized touch. So it is known as what? Crude touch. Crude touch. Okay. While the tectoreceptors and the thigmoreceptors, especially the tectoreceptors, they are sensitive to fine touch. Phonoreceptors sensitive to sound, ear; olfactoreceptors sensitive for smell, nose; gustatoreceptors sensitive for taste, tongue; thermoreceptors sensitive to temperature. Okay. Caloreceptors sensitive to heat, photoreceptors sensitive to cold, galvanoreceptors sensitive to electric current, rheoreceptors sensitive to water or air current, georeceptors sensitive to gravity, telereceptor sensitive to distance. Receptors of vision, hearing, and smell receive stimuli from a distance, hence called telo, teleroreceptors. So teleroreceptors, they are sensitive to distance, like receptors of vision, hearing, smell. They receive stimuli from distance, and hence they are known as what? Teleroreceptors. Proprioceptors, they are located in the skeletal muscles, joints, tendons. It is from these receptors that we know the position of our arm or leg without having to look at it. So proprioceptors, they are located in the skeletal muscles, joints, tendons. It is from these receptors that we came to know about the position of arm or leg without having a look at it. So gustatoreceptors, teleroreceptors, georeceptors, rheoreceptors, eoreceptors sensitive to water or air current. Gustatoreceptors sensitive to taste. Galvanoreceptors sensitive to electric current. Okay. Proprioceptors sensitive to position of the body organ. Okay. Then comes the interoreceptors. Interoreceptors, they are present in internal organs. Example, receptor for hunger, thirst, pain, and balancing, which are placed in hypothalamus. Okay. So these receptors, they are present within the body and they are known as what? Interoreceptors. There are some tangoreceptors, you have to learn the name. Okay. Merkel's corpuscles found in the epidermis, stratum malpighi of skin. Merkel's corpuscle, it is a tangoreceptor, okay, which is respond to touch and pressure. Touch and pressure. Merkel's corpuscles found in the epidermis, stratum malpighi of skin. Merkel's disc found in epidermis, stratum malpighi of skin. So Merkel's corpuscles and Merkel's disc both are present in stratum malpighi of skin. Mesner's corpuscles present in the skin around the base of hair, okay, base of root hair plexus and feather. These are sensitive for touch and pressure both. That is why they are known as what? Tangoreceptors. So Mesner's corpuscles, they are present in the skin around the root hair plexus, okay. Genital corpuscles, they are sensitive cells with nerve endings in skin around the genital organ. Grande's corpuscles found in the birds at the base of the beak, they are kidney shaped in structure. So Merkel's corpuscles, Merkel's disc, Mesner's corpuscles, genital corpuscles, Grande's corpuscles found in the birds at the base of beak, they are kidney shaped in structure. Herbert corpuscles found in the buccal cavity of birds. Herbert corpuscles found in the buccal cavity of birds. Capsulated corpuscles, they are sensitive cells encapsulated and found in skin. Capsulated corpuscles, they are encapsulated sensitive cells which are found in skin. Pacinian corpuscles, important, found in deep layer of dermis and sensitive to touch, pain and pressure. Okay. So Pacinian corpuscles, they are response to strong pressure. Strong pressure. Okay, pain and touch. So for humans, Merkel's corpuscles found in the epidermis, stratum malpighi of skin. Merkel's disc found in the epidermis, stratum malpighi of skin. Mesner's corpuscles present in skin around the base of hair and feather. They are sensitive for touch and pressure both. Then Pacinian corpuscles found in deep layer of dermis and sensitive to touch, pain and pressure. Okay. Then Golgi's corpuscles or Mesonian corpuscles, they are sensitive to touch and found in subcutaneous region. Mesoni corpuscle or Golgi's corpuscle, they are found in the subcutaneous region of the skin. They are also sensitive to touch. There are some important olfactoreceptors. Jacobson's organ, which is known as what? Vomeronasal organ. It is concerned with smell. They were the first appear in amphibians and well developed in snake, lizard, and spinodon, that is a reptile. These are less developed in birds and mammal. Structure is blind sac, okay, and lined by olfactory epithelium, Nerdian membrane. Jacobson's organ is not found in rabbit. In man, it is vestigial organ. In human fetus, Jacobson organ is present. Jacobson organ also found in marsupials, rodents, and insectivores. So Jacobson's organ is found in marsupials, rodents, insectivores. It is also known as vomeronasal organ. It is concerned with smell. They were first appear in amphibians and well developed in snake, lizard, and spinodon. Spinodon is a reptile. They are less developed in birds and mammal. Okay, structure is blind sac. Okay, and they are lined by olfactory epithelium, that is Nerdian membrane. Jacobson's organ, they are not found in rabbit. Okay. Crows and bulb. Crows and bulb, these are sensitive for temperature. So these are thermoreceptors. So crows and bulb, they are sensitive for temperature and pain and frigio in nature. Okay, mean change in temperature. Found in lips, tongue, conjunctiva of eyes, and corium of finger. So crows and bulb, they are sensitive for temperature and pain and frigio in nature. They are found in lips, tongue, conjunctiva of eyes, and corium of finger. Organ of Ruffini, sensitive to temperature and mainly related with heat. Organ of Ruffini or Ruffinian corpuscles, they are sensitive to temperature and mainly related to heat. They are thermoreceptors. Okay. Ampulla of Lorenzini. Ampulla of Lorenzini is found in snout region of cartilaginous fish. These are helpful in detecting the temperature of water. Ampulla of Lorenzini is found in the snout region of cartilaginous fish. They are helpful in detecting the temperature of water. So crows and bulb, they are sensitive for temperature and pain, and they are frigio in nature. They are found in lips, tongue, conjunctiva of eyes, and corium of finger. Okay. Organ of Ruffini, they are sensitive to temperature and mainly related with heat. They are sensitive to temperature and mainly related to heat. Okay. Ampulla of Lorenzini, they are found in snout region of cartilaginous fishes and they are helpful in detecting the temperature of water. So these are important thermoreceptors. Okay. Now comes, we are going to discuss eye. It is a 3D model of eyeball. Okay. Eye is a hollow spherical structure measuring about 2.5 centimeter in diameter. Its wall is composed of three major coats which will be discussed in the coming slides. Okay. So this is the eyeball. You can see eye. Human eye have binocular vision. The eye can discriminate color. It appraise length, width, and depth. Okay. And from true and real and inverted image. Structure of eye. The eyes are two in number and they are lodged in orbits known as what? Eye orbits, which are bony socket of skull. The eye is hollow spherical organ about 2.5 centimeter in diameter and about 6 to 8 gram in weight. It has two parts. So it is 2.5 centimeter in diameter and it is having 6 to 8 gram of weight and it has two parts, two major parts. Okay. One is the protective devices. Eye has four protective devices. One is the eyebrows. They are outwardly directed hair of the eyebrows carry the sweat and raindrops trickling down the forehead to the sides to prevent their falling into the eyes. So eyebrows basically protect the eye from sweat and raindrops which directly trickle down from the forehead, okay, to the sides. Then comes the eyelids or palpebrae. Eyelids or palpebrae in men, two eyelids are present. Upper is movable. They are regularly closed at short intervals to clean the cornea. Okay, this is called blinking of eye or blinking reflex. In frog, out of two, upper eyelid is immovable, and the lower eyelid is movable. In case of frog, the upper eyelid is immovable and the lower eyelid is movable. Nictitating membrane is present in frog which protect the eye in water. Movement of nictitating membrane takes place by retractor bulbi. It becomes folded by levator bulbi. So eyelids or palpebrae, two eyelids are present. Upper one is movable and the lower one is upper one is immovable only. They are regularly closed at short intervals to clean the cornea. It is called blinking reflex. In frog, out of two upper eyelids, okay, the upper eyelids is immovable and the lower eyelid is movable. The lower eyelid is movable. Nictitating membrane is present in frog to protect the eye in water. Movement of nictitating membrane takes place by retractor bulbi and it becomes folded by levator bulbi. So levator bulbi and retractor bulbi. Retractor bulbi. Okay. Folding is done by levator bulbi. It is a non-functional vestigial nictitating membrane called plica semilunaris. Plica semilunaris is nictitating membrane which occur in human eyes. It remains permanently retracted at the inner angle of each eye. Plica semilunaris. Then come eyelashes. Okay. The eyelids they bear at their free edge a row of stiff hair known as eyelashes. They check the entry of dust particles, tiny insects, and raindrops into the eyes. So it is also protective in function. Okay. Then comes the eye glands. Eye glands. The first is meibomian gland. The eyelids bear at their free edge a row of meibomian gland, that is the modified sebaceous gland, which basically act as a lubricant. So meibomian glands they are present at the free edge, okay, of the eyelids. They are modified sebaceous gland. Meibomian gland is a modified sebaceous gland and they act as a lubricant. Lacrimal gland or tear gland. It lies in the upper outer part of the orbit and secretes a slightly saline, means alkaline, watery fluid that contains a bacteriolytic enzyme lysozyme. This secretion moistens the surface of the eyeball. The excess of this secretion passes through the nasolacrimal duct and it is a modified sweat gland. So nasolacrimal duct is, or we can say the lacrimal gland or tear gland is a modified sweat gland and meibomian gland is a modified sebaceous gland. Please remember, meibomian gland is a modified sebaceous gland and lacrimal gland is a modified sweat gland. Okay. Harderian gland. Some aquatic mammals like whale possesses Harderian gland which lubricate the nictitating membrane. It is also found in frog and birds. So Harderian gland is present in mammals, aquatic mammals like whale, okay, which lubricates the nictitating membrane, okay. It is also found in frog and birds. Glands of Zeiss. These are modified sebaceous gland. Glands of Zeiss are modified sebaceous gland, okay. Meibomian gland is also modified sebaceous gland. Okay, they are found at the base of hair follicle of eyelashes. Poor lubricating fluid in hair follicle. They pour basically. They pour lubricating fluid in the hair follicle. Infections of these glands is known as what? Style. So glands of these, they are modified sebaceous gland. They are present at the base of hair follicle of eyelashes. They pour the fluid in hair follicle. Okay, and infections of these glands is known as what? Style. Glands of Moll. It is a modified sweat gland. So glands of Moll is a modified sweat gland and open into the follicles of eyelashes. So glands of Moll is a modified sweat gland and it's open into the follicles of eyelashes. In human, Meibomian gland, lacrimal, Moll's gland and Z's glands are present. So in humans, Meibomian gland is also present, lacrimal gland is present, Moll's gland is present and Z's gland is present. Hardarian gland is not present in humans, it is present in frog and birds and aquatic mammals like whale. Okay. Now this is the structure. Okay. The outer coat, you can see the outer coat which is fibrous known as what? Sclera or cornea. Okay. So you can see this is sclera, which is a this is sclera, which is outer fibrous coat. Okay. The middle coat is choroid. This middle coat is choroid. It mainly contains blood vessels. Okay. So the middle coat is choroid, which mainly contains blood vessels. This middle coat is the vascular coat. So the outer coat is fibrous. Middle coat is vascular, and the inner coat is nervous. So the outer fibrous coat is sclera, the middle vascular coat is choroid, and the inner coat is nervous, which is retina. The middle coat, which is vascular, it consists of choroid and ciliary body and iris. Okay. Now you can see the fibrous coat is further divided into sclera and the cornea. This is the cornea. Okay. So starting from the cornea, the outer portion you can see the bulbar conjunctiva. Okay. Then cornea. Then this is pupil. Okay. This is pupil. This is the muscular region is iris. Okay. The circular muscles of iris. This is lens. These are suspensory ligaments. This is the sclera, the outer coat. This is the median rectus muscle or medial rectus muscle. Okay. The retina supplied with arteries and veins. Central retinal artery, central retinal vein. The outer is dura mater. And here comes the optic nerve, which is the second cranial nerve. This is optic disc, which is known as what? Blind spot. Okay. The choroid is spatially thick in the muscular region. So in the muscular region, the choroid is thick. This is fovea centralis of macula lutea. Okay, here, or blind spot. The blind spot is optic disc. Okay, and this is the fovea centralis macula lutea, which is the yellow spot. Okay, which lies on the visual axis. Okay, yellow spot. Okay, this is vitreous humor in the posterior chamber or the vitreous body. Okay, this is the lateral rectus muscle. This is ora serrata. Okay, this is the ciliary body, which is having mainly two parts: the ciliary process, which gives to secrete the aqueous humor, and these are the ciliary muscles. Okay, then comes the sclera. Okay, canal of Sheen, and then the anterior chamber and the posterior chamber. Okay. Now, Now comes the different layers. The first layer is sclerotic layer or the fibrous tunica. Fibrous tunica is further divided into sclera and cornea. It is the outermost and opaque. It is fibrous and non-vascular layer, easily seen as white part of the eye. White part of the eye. It is a coat of the dense connective tissue. It is mainly made up of dense connective tissue, which is made up of collagen fibers and fibroblast. So sclerotic layer or the fibrous tissue, it is the outermost part which is opaque and non-vascular. Okay, it can be easily seen and identified as the white part of the eye. It is a coat of dense connective tissue made up of collagen fibers and fibroblast. Sclera covers the entire eyeball except cornea. Except cornea, sclera covers the entire eyeball. Gives shape to the eyeball. Okay, it gives shape to the eyeball. Sclera in frog is cartilaginous. Very important. In case of frog, the sclera is cartilaginous. Okay. It protects and maintains the shape of the eyeball. Okay. Then comes cornea. Cornea is a transparent portion that forms the anterior 1/6 of the eyeball. Okay. It is the transparent portion, okay, which forms the 1/6 part of eyeball. So cornea, it is in the center. It is a sclerotic layer only. Okay. It merges with the transparent round window called cornea, which makes the 1/6 part of the eyeball. Okay. The cornea admits and helps to focus the light waves as they enter the eye. The cornea is avascular, gets no blood supply. It is avascular, gets no blood supply. Okay. This part of the eye absorbs oxygen from the air. The cornea was one of the first organ to be successfully transplanted because it lacks blood vessels. Okay. At the junction of sclera and cornea, there is a structure called canal of Schlemm. Canal of Schlemm. From the anterior chamber, the aqueous humor from anterior chamber, which is continuously produced and is drained off into the canal of Schlemm. Drain into canal of Schlemm. Okay. Then comes the conjunctiva. The cornea and the exposed part of the sclera are covered externally by a thin transparent membrane called conjunctiva. Okay. Choroid layer, which is the vascular part. The vascular coat consists of choroid, ciliary body, and iris. So it is known as vascular tunica, also known as uvea middle. Known as uvea middle. It is a vascular layer which supplies the nutrients to the eye. It is distinguished into three parts: choroid, ciliary body, and iris. So the choroid layer, which is vascular tunica, okay, it is also known as uvea middle. It is vascular layer which supplies nutrients to the eye. It is distinguished into the three parts: choroid, ciliary body, and iris. Choroid is highly vascular, posterior portion of the vascular tunic. So basically the vascular tunic is the middle vascular coat and choroid is highly vascular and posterior portion of it. The choroid occurs in the main part of the eyeball adhered to the sclerotic layer. So choroid basically occurs in the main part of the eyeball and it is attached or adhered to the sclerotic layer. The pigment is reddish in rabbit and black and brown or bluish in man. So the pigment in case of choroid, okay, is reddish in rabbit and black or brown or bluish in man. So this is the choroid layer. The choroid, it lies just adjacent to sclera and contains numerous blood vessels that supply nutrients and oxygen as we have discussed. Okay, that is why it is known as what? The vascular coat. Okay, it contains different pigmented cells that absorb light and prevent it from being reflected within the eyeball. Ciliary body. Ciliary body is vascular and pigmented like choroid, made up of ciliary process and ciliary muscles. The muscles in ciliary, these muscles are only circular type. Okay, no radial muscles. So ciliary body is vascular and pigmented like choroid. Okay, and it is made up of ciliary process and ciliary muscles. Okay, the ciliary muscles are only circular type. Ciliary body, especially when we talk about the ciliary muscles, these ciliary muscles and ciliary processes, ciliary muscles are circular type, ciliary processes, they secrete aqueous humor. Secrete aqueous humor. Okay, the ciliary muscles are smooth muscles. Ciliary muscles are smooth muscles, and they are circular muscles. Okay, so the smooth muscles, in case of ciliary muscles, they are of two types, circular and meridional. Okay, but mostly circular muscles are there. Okay, attached to the ciliary body. So these are attached to the ciliary body, and they are known as suspensory ligaments. Okay, the ciliary body is basically hidden by iris. The ciliary body helps in the accommodation of altering the focus of eye from object or shape of the lens for near vision and far vision. So the ciliary body is basically hidden by iris and it helps in the power of accommodation. Power of accommodation. Iris. Beyond the ciliary body, the vascular tunic sharply turns inwards, forming a circular shelf-like diaphragm called iris. Okay. The color of the iris is responsible for the color of the eye: brown, black, blue, or green. In albinos, iris is deficient of pigments. Okay. So iris is circular muscular diaphragm. Iris is circular muscular diaphragm containing the pigments which give color to the eye like black, brown or green. In albinos, iris is deficient of these pigments. Okay. Now, iris basically separates the aqueous humor region into anterior and posterior chambers. So iris, it separates aqueous humor into anterior region and posterior region. Iris extends from the ciliary body across the eyeball in front of the lens. Okay. It has an opening in the center called pupil. So iris only basically extends from the ciliary body across the eyeball. You can see here the iris. This is the iris, which basically extends from the ciliary body from the across the eyeball in front of the lens. In front of lens. It has an opening in the center called pupil. So the opening is known as what? Pupil. Okay. It contains two types of smooth muscles. Two types of smooth muscles. First is the circular muscles, which are known as what? Sphincters. And the second is radial muscles, known as dilators. And they both are of ectodermal in origin. Okay. The iris controls the amount of light entering the eye. Okay, it contains the amount, controls the amount of light entering the eye by radial muscles entering the eye by the help of radial muscles contracting in dim light. So, because of the contraction of radial muscles, okay, especially in dim light, and the circular muscles contracting in bright light. Contracting in bright light. Okay. So this set of circular muscles and the what we can say radial muscles. Okay, they are under the control of autonomic nervous system. They are under the control of ANS, autonomic nervous system. Okay, the sympathetic stimulation causes the radial muscles to contract. When sympathetic stimulation is there, radial muscles contract. Okay, and the pupil is dilated. Pupil dilates. Okay. Parasympathetic stimulation causes circular muscles to contract. Circular muscles contract. And the pupil is constricted. The activation of sympathetic nervous system, okay, helps to contract radial muscles and pupil gets dilated. And in case of parasympathetic nervous system activation, the circular muscles contract, which helps in the constriction of pupil. Okay. Lens. Lens is the colorless, transparent, and fibrous crystalline structure made up of protein, okay, 1 and 2 crystalline protein and enclosed in lens membrane. So basically these two crystalline proteins, crystalline protein 1 and 2, they are enclosed in the lens membrane, okay. So lens is a transparent, colorless and fibrous structure, but it is crystalline and it consists of crystalline proteins. Understood? The lens is ectodermal in origin. Lens is basically ectodermal in origin, okay? And it is lodged, it stays in the eyeball by the suspensory ligament of ciliary body. So suspensory ligament helps to hold the lens in their position. These suspensory ligaments, they are the extensions of ciliary body. Lens is ectodermal in origin. It is transparent. It is biconvex. Biconvex. Okay. Elastic structure which bends light waves as they pass through its surface. Okay. The lens the lens mainly consists of, different types of crystalline proteins. As we have discussed, it is ectodermal in origin. Okay. It is composed of epithelial cells that have large amounts of clear cytoplasm in the form of fibers. The capsule present in the lens is composed of different layers of intracellular protein, as we have discussed, intracellular protein 1 and 2. The lens can change shape from movement to movement, and by doing so, it changes the power of accommodation. Okay. The lens can also change shape from year to year, thereby accounting for changes in vision. Suspensory ligament is also called zonula of Zinn. In men, lens is biconvex, while frog it is elliptical or subspherical. Okay. In case of frog, the lens is elliptical or subspherical, while in man it is biconvex. Suspensory ligaments is known as what? Zonologue. Lens divide the eyeball into two chambers. The outer aqueous humor partially divided into anterior chamber and posterior chamber. The anterior chamber is large and the posterior chamber is small. It is filled with aqueous humor, which is mainly watery. Okay, the aqueous humor is formed by the ciliary process of ciliary body, and the inner is vitreous humor or vitreous chamber, which is filled with vitreous jelly or Watson's jelly. Okay, containing 99% of water and some salts. Some salts. Okay, little of mucoprotein, that is vitrin, and hyaluronic acid is also present in the vitreous humor. So aqueous humor is mainly consist of water, okay, while the vitreous humor, okay, or the vitreous jelly or the Watson's jelly, it contains 99% of water. Along with it, it contains salts, okay, hyaluronic acid and mucoprotein vitrin. This is the structure you can see, okay. In this structure, they have mentioned about the ciliary muscles and the iris muscles and their placement. This is the cornea. This region is the aqueous humor or the aqueous chamber. Okay. The posterior surface of the cornea, epithelium of cornea, anterior or elastic lamina, posterior elastic lamina. Okay. Aqueous chamber or anterior chamber, it is given. Aqueous chamber. Okay. This is iris. Okay. The iris is having circular. These are the radial muscles of iris. Radial muscles of iris. These are radial muscles of iris. Okay. The ciliary muscles, they are mainly of circular type. Okay. But they are some of them are meridional as well. So circular muscles and meridional muscles. Okay. Circular muscles, ciliary muscles and meridional muscles. Okay. Now comes the retina. Retina is a neurosensory part known as tunica neurosensory. Okay, it is the innermost, thin and transparent purplish red due to the presence of eye pigment rhodopsin in rod cells and visual purple, which has extracted by Cohen in 1876 and named by Schupperper visual purple. Made It is made up of four distinct layers. So retina is neurosensory tunica. It is the innermost layer which is thin and transparent. It is purplish red due to the due to the presence of rhodopsin, which is visual red in rod cells, and visual purple, also known as visual purple. So eye pigment rhodopsin in reds, also known as visual purple, which was extracted by Cohen in 1876 and named by Schupperper visual purple. Okay. So the rod cells have rhodopsin and the cone cells have iodopsin. Okay. It is a neural coat and sensory layer of eyeball. Its external surface is in contact with the choroid. So retina external surface is in contact with choroid. Choroid. And its internal surface is contact with vitreous humor. Okay, a small oval yellowish area of the retina which lies just opposite to the center of the cornea is called macula lutea or yellow spot. Okay, which lies on the visual axis. Okay, so it is basically containing cuboidal pigmental epithelium towards choroid. Okay, external surface. It consists of layers of rod cells and cone cells, layer of bipolar neurons, okay, layer of ganglia towards the vitreous chamber. Okay, the innermost layer is towards the vitreous chamber and the outermost layer is towards the choroid. Okay, this is given. The pigmental epithelium is the this is the pigmental epithelium. Okay, just a minute, I'll change that. This is the pigmental epithelium. Okay, the choroidal layer. Okay, here you can see the choroidal layer is the outermost, okay, and the vitreous layer or towards it is the innermost. Okay, the pigmental epithelium choroid is the outermost. Then it contains pigment cells, which are photoreceptor layers. This is the photoreceptor layer. This is the outer limiting membrane. This is the outer limiting membrane. Outer nuclear layer. This outer nuclear layer basically consists of rod and cone cells. Okay, their cell bodies. Then outer plexiform layer which contains the axonal parts. Okay, axonal endings. Okay, then the inner nuclear layer. Okay, then inner plexiform layer, the outer plexiform layer, and the inner plexiform layer. So these are the bipolar neurons. You can see the bipolar neurons. Okay, these are bipolar neurons. So this is a bipolar cell. Okay, these are the amacrine cells. You can see the amacrine cells. This is the horizontal cell. Mueller cell, which is a supporting glial cell. These are the Mueller cells or the supporting glial cells. Then this is the ganglionic cell layer and the nerve fiber layer. This is the 10 inner limiting membranes, axons at the surface of retina passing by optic nerve, chasmata, and track to lateral geniculate body. Lateral geniculate body. Okay. So these are the layers. Okay, starting from the pigmental epithelium, then layer of rod cells and cone cells. Okay, means the photoreceptor layer, then the external limiting membrane or the outer limiting membrane, then outer nuclear layer, okay, or external nuclear layer, then the external or outer plexiform layer, okay, then the inner nuclear layer, then inner plexiform layer, then ganglionic cell layer, and the nerve fiber layer. Okay, now we will discuss each layer. The first thing we have to discuss is pigment cell layer. It lies close to the choroid. It consists of single layer of cells containing pigment. Containing pigment. These pigment cells, they are rectangular, okay, or rectangular in vertical section. Their width being greater than their height, okay. So the cells they are having more amount of width in comparison to their height, okay. These cells give rise to pigmented processes or projections extending into the next layer, okay. They have pigmented rectangular cells. Okay, having more width, and they also have pigmented processes of projections. Then the second is layer of rod cells and cone cells. The rods are the process of rod cells and the cones are the process of the cone cells. Total number of rods in human retina have been estimated to 110, 100, I was about to tell you 115, but it's okay. 110 to 125 million. And the number of cone cells 6.3 to 6.8 million. The rods contain a photosensitive pigment called rhodopsin, also known as visual purple. Visual purple. Rhodopsin is composed of So rhodopsin is basically the visual purple. Rhodopsin is composed of opsin and retinin. It is composed of yes, opsin and retinin. The opsin is a protein and is called scotopsin. Scotopsin. Opsin is a protein and is called scotopsin. Okay. The retinin is an aldehyde of vitamin A, retinal, aldehyde of vitamin A. Okay. And is called retinal. Okay. The rods mainly enable the animal to see in dark. Help to see in dark. Okay, dim light. Help to see in dark. Okay. Therefore, rods are present in large number in nocturnal animals. Large number in nocturnal animals. Okay. The photosensitive pigment in cones is of three types. Three types of pigment. The first is porphyrosin, then idopsin, and synopsin, which give response to red, green, and blue light, respectively. Red, green, and blue light, respectively. Okay. The responses to different colors basically is produced by the different combinations of these three types of cone cells and their photopigments. Okay. When the three types of cones are stimulated equally, a sensation of white light is produced. So when they are equally stimulated, then sensation of white light is produced. The protein in cone pigment is called photopsin. Photopsin is the protein. In rod cells, opsin is the protein. Here, photopsin is the protein. Okay, is that clear? Now, the third layer is external nuclear layer. It mainly contains of cytons or cell bodies and nuclei of rod cells, rod and cone cells. Okay. Then comes the outer or external plexiform layer, which is having the outer synaptic zone. Okay. This layer consists only of nerve fibers and form plexus, means a network, a mesh-like network of different nerves. The axons of rods and cones synapse here. Okay. The axons of rods and cones get synapsed here, okay, with the dendrites of bipolar neurons. Then comes internal nuclear layer. It consists of the cell bodies of bipolar neurons, horizontal neurons, and amacrine neurons. Cell bodies. Okay. Then comes the internal plexiform layer or inner plexiform layer. Okay. Also known as inner synaptic zone. Here the synapse of the synaptic nerve fibers of bipolar neurons, ganglion cells, and amacrine cells are there. It is also known as internal plexiform cells. These cells are known as what? Internal plexiform cells. Then comes the layer of ganglionic cells. They contain cell bodies of ganglions. Then the axons of these ganglion cells, they merge to form layer of optic nerve. Axons of ganglionic cells. So layer by layer we have discussed to form optic nerve, which is the second cranial nerve connected to the brain. The nerve fibers from all the parts of retina converts to leave through a blind spot. So basically, there is a region known as blind spot or optic disc. So it is the portion where all the nerve fibers from retina they converge to leave through a space known as what blind spot, which contains no rods and no cones. And therefore there is no image formation. That is why it is known as what blind spot. Then comes the retinal gliocytes. These retinal gliocytes are known as what cells of Mueller. Okay, as seen in the picture, I'll show you. Mueller cells supporting glial cells. Okay, so in addition to bipolar neurons, horizontal neurons, and amacrine neurons, the internal nuclear layer also contains the cell bodies of retinal gliocytes. Contains cell bodies of retinal gliocytes or cells of Mueller. These cells form numerous protoplasmic process extending through the external and or we can say the internal limiting membranes. So they merge to the external or the internal limiting membranes. Protoplasmic process limiting membrane. The internal limiting membrane separates the retina from vitreous humor. Separates retina from vitreous humor. It is assumed that retinal gliocytes they cover up the neurons of retina, okay, and they also serve as nutritive in functions, okay. So this is the different types of layers of retina. Fovea centralis, a little part of retina that lies upon the optical axis called area centralis. Here the retina is very thin and contains only cone cells filled with yellow pigment. That is why the name lutea is given, macula lutea. This part is called yellow spot of macula lutea. In man, rabbits and other mammals, but not in frogs, this area has a small shallow depression called fovea centralis. Okay, so fovea centralis is present in man, rabbits, and other mammals, but not in frogs. Okay, the latter is the most sensitive part of the eye, the area of most acute vision. Okay, so fovea centralis is the area of most acute vision. It is also claimed that cone cells and fovea centralis are placed somewhat obliquely. Okay, so that these can form magnified images of the object. They can form magnified images of the object. Okay, blind spot we have already discussed. Difference between aqueous humor and vitreous humor. Aqueous humor is a watery fluid. Vitreous humor is a jelly-like substance. Aqueous humor is present between cornea and lens. Okay, between cornea and lens. And vitreous humor is present in lens and retina. Between lens and retina. Secreted continuously by ciliary process and drained in canal of Schlemm. Okay, they are not replaced. Okay, obstruction in the flow of aqueous humor may damage retina. By increasing intraocular pressure and is the reason of glaucoma. Okay, and vitreous humor, it does not flow. No flow is seen. So, ladies and gentlemen, we have completed till the difference between aqueous humor and vitreous humor. Okay, so there are some differences more for rod cells and cone cells, adopsin and rhodopsin, the muscles, the accessory structure of the eye, and the especially the functioning of the vision. Okay, power accommodation, functioning of the vision, mechanism of vision. Okay, everything will be discussed in the next lecture. Okay, till then, bye bye, take care, stay safe, stay healthy.